Today's presentation is Bat Winter Roosts in Texas by Dr. Missy Meyerhofer. I'm excited to get to introduce her. And in 2015, Texas Parks and Wildlife contracted with the Natural Resources Institute at Texas A&M University to conduct a large-scale statewide survey of bat winter roosts in Texas. At the time, white nose syndrome was quickly spreading towards Texas, and we viewed this as the last opportunity to document bat populations at winter roosts before impacts from white nose syndrome took place. Missy became the lead on this daunting and quite large project, and I was very impressed by her persistence and determination to see it through. Dr. Meyerhofer is a postdoctoral researcher in Bat Lab Finland under the direction of Dr. Thomas Lilly. She is focused on understanding diseases in European bats, and Melissa completed her PhD at Texas A&M University in the Wildlife and Fisheries Science Department under the direction of Dr. Morrison, while working as a research associate for the Natural Resources Institute on this project that she'll be presenting on today. She also completed a master's in animal behavior at Bucknell University, Pennsylvania, where she studied physiological consequences of white nose syndrome under the supervision of Dr. Reeder, and she obtained a bachelor's in arts in physiobiology and studio art from Ripon College in Wisconsin. And so with that, it's my pleasure to get to introduce Missy to this webinar, and I'm going to stop sharing my screen and pass controls over to her. And just
all right, let's see.
I can hear you, Missy.
Perfect. I'm trying to get to the share. Well, I'll just share my screen. Do the whole thing. Yep. Great. There we go.
Perfect. That's great.
Everyone for coming to this talk. And I hope you all have a great lunchtime. It is currently 8 p.m. in Finland. And today I will be talking about my 4-year project, which involved trying to get some baseline information on overwintering bats and their roost environments in Texas. So for an overview of this talk, as it is going to be about 40 some odd minutes, I'm going to give you a background and the reasoning as to why this project was so important to do. And then I will go into a few projects that have been published, one focusing on understanding the winter habitats of 7 of the bat species that we documented in Texas there in the winter, as well as predictors of presence and the abundance of tricolored bats occupying culvert roosts in Texas. And finally, I will just touch briefly on a disease spread model that I've been working collaboratively on with folks here in Finland. So the real big reason why we were looking at trying to gather all this baseline information in Texas was because of the disease of bats known as white nose syndrome. White nose syndrome is caused by the cold-adapted fungus or cold-tolerant fungus known as Pseudogymnoascus destructans. It's a mouthful, so I'll keep it to PD throughout the talk. It was first documented in North America in 2006 in a cave and has since spread up into the northern parts of Canada, as well as down and westward across the United States. This fungus, as you can see, is named, and the disease is named for the characteristic features that you see on this bat, the white fungal growth on the muzzle, ears, and wing membranes of some hibernating bat species. So unfortunately, this disease has caused precipitous bat declines of some of the hibernating bat populations in North America. In particular, a couple of the species that were most greatly impacted are the little brown myotis, as well as the tricolor bat. And so it's a really devastating disease in that some of these colonies have been nearly wiped out, whereas others have been wiped out. But what's also interesting about this disease is that it doesn't impact all bat species the same. There's a lot of differences based on where they choose to roost, how big the bat is, how long their hibernation period is, and several other factors. And as I'd said, this fungus is cold adapted. It grows between about 0 and 19.8 degrees Celsius, which is in upwards about 69 Fahrenheit, but it optimally grows right around the 12.8 or 12.5 to 15.8 degrees Celsius range. And unfortunately for bats, bats that overwinter in roosts such as caves are choosing environments that fall within these temperature ranges. Bats tend to hibernate or use torpor between temperatures of about 0 and 10 degrees Celsius. So when you have a bat occupying this environment that is conducive to the growth of this fungus, you end up potentially getting the disease. And as I'd said, the fungus, which originated and first documented in New York, has spread into Canada, across the United States. Unfortunately, this fungus is spread bat to bat. It can also be spread from the environment to the bat. And it is believed to have been spread from Europe and Asia into the United States from a human. So there are various different ways that this fungus can spread. Now, what you don't see on this map is that all of these counties are kind of following the karst regions, which are regions where can form. And this makes sense because where the caves are, you're most likely to have bats. Bats are using some of these caves during the winter, and then you get development of the disease. So you can see how it is spread across the United States. And this is when I started the project in November 2015. We didn't have any knowledge of what was happening in Texas. We had this idea that, yes, the fungus is going to spread through Texas. There's a plethora of caves that span from the Texas Panhandle, the northern part of Texas, down into central Texas, and then across Texas where you get Carlsbad Caverns, which reaches into New Mexico. And in addition to that, in Texas, we have all these different bat species. So we didn't have an idea really where all the caves are, how many bats are in these caves, and what are the environments like. But I will say that the caving community did have a lot of this knowledge, as well as other researchers, but we didn't have any really relevant and recent knowledge that we can use to really help us assist in pinpointing certain areas to really monitor and manage for different bat populations. So in Texas, Texas is a pretty cool state for bats. In the United States, there are several different bat species. However, in Texas in particular, we have 33 bat species. These species exist, some of them in the eastern United States, as well as in the western United States. So we have this overlap of different species in Texas. And at the onset of the study, we knew that several species, such as the northern long-eared bat, the big brown bat, and the tricolored bat, could be affected by white nose syndrome. And we knew that from other states in Northeast United States. We also knew that other bat species could just be carriers of the fungus. And so the difference between a carrier and a bat that gets white nose is that you see that white fungal growth, you have dead tissue, or you see emaciated or really sick looking bats, or unfortunately you find dead bats. Whereas bats that are carriers, we can document the fungal DNA on the bat, but they don't seem to show any signs of getting sick from the disease. So the approach to this 4-year-long project, trying to cover the entire state of Texas, which if you don't know how big the state is, from one site to another, I think the farthest I drove was about 9 hours. We needed to cut down the size of Texas for sampling. So we used the North American Bat Monitoring System, which is comprised of a grid layer, which is 10 by 10 kilometer grid cells. We overlaid that over Texas and then randomly selected grid cells for sampling. And we purposefully biased our sampling to northern Texas and northeastern Texas, where we anticipated the first documentation of the fungus based on the closest locality of where we knew white nose syndrome was and where we knew the fungus was present. We then reached out to various different entities, such as the Texas Department of Transportation, who provided us with a data set of all the culverts and bridges in the state of Texas, which just so happens to be greater than 60,000 different potential locations. We then reached out to the Texas Speleological Survey for information on caves as well as other cavers to try to identify and gain access to those sites. We worked collaboratively with Bat Conservation International, who was surveying caves in northern Texas. And we also worked with folks such as the Texas Master Naturalist, other researchers and biologists in the state. So once we had all of this information and we continued to gain access, when we got to a site, would enter the site, count all of the bats that we could visibly see and identify them to species. And thankfully, within the state of Texas and for the species we were seeing, they were all morphologically distinct. So we didn't have to disturb them any more than we already were by being in those sites. We then swabbed a subset of bats to test for the fungal DNA, and we used kits that were provided by the National Wildlife Health Center, provided by Dr. Winifred Frick, and as well as kits that we created ourselves to try to sample and swab as many different bats and bat species across the state. And finally, we collected some environmental characteristics and structural characteristics of these sites. So at a certain subset of caves, we collected temperature and humidity year round, and we did this at culverts as well. And then we also collected some information about where the bat was roosting. So here's an image of some of my former teammates. They're in a cave in the northern Panhandle region in a gypsum cave. The bats that you're seeing are all cave myotis. They are currently counting the bats, identifying them, and also checking them for any growth of the fungus. And we did this at every site that we had access to. And then we also swabbed, as I said, a subset of bats. So we, as carefully as possible, would take a Q-tip swab that was sterile and rolled it over the muzzle of hibernating bat species. So as you can see here, my former technician is currently swabbing a tricolored bat. And here's a close-up image of the tricolored bat. And as I said, we did this for a random selection of bats. Typically, they were ones that we could more easily access, but we tried to swab bats throughout the site to get an idea of whether or not the fungus was present in any part of the site, be that a cave or a culvert. And so this brought us to a lot of really unique locations. And as I said, without all the collaborative efforts with all the landowners in the state of Texas, as Texas is predominantly privately owned, 97% of the land, we wouldn't have had access to a lot of these different locations. And so this is an image of a cave that was in central Texas in the backyard of a landowner. And so some of these caves became very complex, and we did our best to try to survey the entire site. And other caves were also quite interesting. This is an image of a gypsum cave in the Panhandle region, and you can see that there's a presence of water. So when we came to these sites, we obviously had to modify how we were going to go about surveying the sites. And we also collected all of this information. So whether or not water was present could also be an indicator of whether or not these bats had a higher risk of getting the disease. And at some sites, we also had multiple different species. This image is just of cave myotis, but in this cave in particular, there were multiple different species occupying multiple different regions of the cave. And like I had said previously, some of these caves and other sites were just massive, miles long. And so we did our best to survey at least a section of each site to just get an idea of what was happening. And in addition to all these different cave locations, we also went out and surveyed under culverts as well as bridges. And culverts, for your information, are drainage systems that are under roadways. So we spent a lot of time in the ditch surveying these different culvert and bridge features. And so we had some information based on historic surveys and from Texas Department of Transportation that there were some historic colonies of tricolored bats occupying culverts. And so we surveyed those as well as all of the random bridge and culverts that we selected using our random selection. And we surveyed many different types of bridge designs. The image you're seeing here is of a parallel box beam bridge. It is a design that bats tend to prefer. And you can tell that they prefer this because they really like to cluster in tight crevices. And this bridge is ideal because of the tight crevices that you are now seeing. And so we surveyed several different locations with this design, as well as a lot of different bridge designs across the state. And finally, we did have some information on some other sites, such as parking garages and bat boxes and alternative different roosts. And one in particular that I thought was kind of interesting that we got information on was a tree roost where we had southeastern myotis occupying. So to survey this one, you just had to lay down and put your head in the hole, look up, and try to count as best as you could the bats that were occupying this tree. And so that was easily said, not as easily executed, but with all the help, we managed to survey over 600 different locations across the state of Texas over the 4 different years of surveying. Oh, and before I go on, I will say that you can tell that there was definitely more survey efforts in northern Texas and northeastern Texas, where we have more of the caves in the Panhandle. Central Texas had caves as well. Eastern Texas had a couple, but were mainly the culverts and bridges. And then we had culverts and bridges in other sites throughout. And after all of those surveys, we identified these different species at different caves during the winter. So our winter was between about mid-March or mid-October through about mid-March, sometimes a little bit later, and sometimes we ended a little bit sooner. In Texas, the temperatures can be pretty warm year-round, so it's very hard to identify a winter period. But we had the Mexican free-tailed bat, the cave myotis, tricolored bat, big brown bat, southeastern myotis, Raffinus and Townsend's big-eared bats, and the ghost-faced bat. And as I'd said at the beginning of the study, we knew that the tricolored bat and big brown bat were 2 species that could be affected by white nose. And it was also initially thought that the cave myotis was. They retracted that information, but then we determined that they are susceptible to the disease. And this makes sense because myotis species tend to be more susceptible than not. And during our surveys, we documented Mexican free-tailed bats with presence of the fungus, as well as the Raffinus big-eared bat and southeastern myotis. So briefly through the swabbing, I'm not going to go into the number of sites, but in year 1, we did not find any presence of the fungus. Also in the first year, as we were just starting this, we had very few sites because we were trying to obtain access and information on different locations. So it made sense that in years 2 through 4, we continued to get positive sites. Our first identification of the fungus was up in the Panhandle. And as the years went on, we found more and more sites returning positive for the fungus, as well as more counties in Texas. So, as I said, the first year or the first winter that we detected the fungus was in the spring of 2017. We had 6 different counties in the Texas Panhandle, which was kind of what we thought where we'd find the fungus. And as time went on, we added a couple more counties in the Texas Panhandle, as well as now in Central Texas. And that was at its lowest latitude that we'd ever documented this fungus in North America. And then we added even more counties spread across Central Texas where we had presence of the fungus. These sites were predominantly cave sites, but we also had a tunnel and a bridge that resulted in positive results. And so to get into one of the studies that resulted from some of this data, as we saw this progression of the fungus, it was obviously apparent that we needed to get more information about the particular habitats and microclimates that were being used by the different species that we identified during the winter. And so the goal of one of the studies that we published was to describe the various microclimates used by 7 different bat species as hibernacula across the state. And so these are our individuals. They were all the previous ones except for the ghost-faced bat that we had information on within their known range across the state of Texas. And so our data included 97 different hibernacula. So this was caves, culverts, tree roosts, and bat towers. It spanned over 6 different ecoregions. And so I haven't talked about ecoregions previously, but this is just how the state is divided by its different environmental features. And we had the 7 bat species. And with these 7 different bat species at all these different sites, we collected air temperature and vapor pressure deficit, which is just another way of looking at the humidity at the site. And we did this right near each of the individual bats. And in addition to that, we recorded the bat's skin temperature. So it was pretty simple. We just used this temperature and humidity pen to grab the temperature near the bat. And we also used a temperature gun, and we took the temperature of the bat as well as the substrate to show that these bats were within a degree of the substrate. And so now you can see the different ecoregions divided out on this map. And our results are pretty straightforward and easy to discuss. So in northern Texas, not surprisingly, the sites in general were colder than the ones found in central and southern Texas. And we also noted this within species. So in particular, we had cave myotis occupying northern and central Texas. And we saw that within the species, they were selecting sites that were cooler in the north than in the central regions of Texas. And I think this is one of the most interesting and important figures for managers and to get a basic understanding of what species might be affected by the disease. So on the bottom, we have our codes for the different bat species, which I will get to in a minute. And along the left-hand side, we have at the top for figure A, the average roost temperature, so the air temperature near the bat, the average skin temperature of the bat. And then for figure C, we have the average VPD, which is vapor pressure deficit. And so when we look at the temperatures that they're choosing to roost at, the air temperature and their skin temperature, and going back to the knowledge that the fungus grows best between 0 and 19.8, we noted 5 different species that were overwintering in Texas that fell within that range. And that's the Townsend's big-eared bat, the big brown bat, the southeastern myotis, the cave myotis, and the tricolored bat. And we also had, if you remember, these environmental data loggers that were recording temperature year round. And so we pulled that information in, and we pulled in information from PRISM, which is a climate group that pulls in temperature information on the landscape. And so we can look at the relationship between the external temperature and the internal temperature of these different sites, and also compare that to the growth temperatures of the fungus. So at the top, you see two different caves, and below you have two different culverts. And the line that is more smooth in all of these represents the internal cave temperatures, and the one that fluctuates a little bit more is external. And internal and external temperatures did correlate. So as the external temperature increased, so did the internal cave temperature, for example. And you're also looking at a parallel gray box, and that is where the optimal growth temperatures are for the fungus. So what you might notice is that at some points for these sites, the temperatures are falling within the optimal growth range of the fungus. And then when we overlay just the general growth range of the fungus, that 0 to 19.8, we see that for a majority of the winter period, these sites are within the growth range of the fungus. And also for your information, these 4 sites were also occupied by bats during the winter. And so for discussion of this, we documented that there is variability in different hibernacula in Texas, with sites in the north being colder than sites in central Texas. However, we did show that a lot of these sites were falling within the suitable ranges for growth of the fungus. And in addition to that, they on occasion fell within the optimal growth range. We noted 5 species that may be affected by white nose syndrome in Texas: the Townsend's big-eared bat, the big brown bat, the southeastern myotis, the cave myotis, and the tricolored bat. And so again, a lot of these species make sense. We already knew the big brown bat and the cave myotis and the tricolored bat could be affected by white nose. We had some inclination to believe that the southeastern myotis would be affected. But one species of interest that we weren't sure about was the Townsend's big-eared bat, and we found this in a cave more in central Texas. And so it was interesting to find out that this species has kind of a lower temperature range, as well as the warmer temperature ranges that we already knew about. And so we can use all of this information to help us guide management of potentially the development of white nose for these species. So instead of trying to manage for all of the different overwintering bats, we can now focus in on certain species based on the temperatures that they are choosing to roost at. And so this kind of brought us to another interesting study focused just on the tricolored bat. During my work, the tricolored bat was petitioned to be listed as a threatened or endangered species. And we had some knowledge of these bats occupying culvert features in Texas. And not only were they occupying culvert features, they were actually more localized in these culverts than they were across the state. Indeed, at least greater than 75% of the known tricolored bat populations from all of the surveys were occupying culverts. And so we had this idea that obviously culverts may be an interesting hibernacula to then look more into. And so we wanted to identify structural and environmental predictors of tricolored bat presence and abundance in Texas culverts. And transportation infrastructure is kind of interesting. It could have variable use by bats, but also in the state of Texas, where typically you find these caves in northern and central Texas, occasionally you have a couple in eastern Texas, culverts become kind of an alternative roost site in regions where caves can't form. And so they could become this critical habitat specifically for tricolored bats, but alternatively also for a couple other species. And so if we could figure out what types of features are driving the selection of these culverts, perhaps if we built those specific culverts, we might get more bats occupying these spaces. And this is kind of interesting because management is just easier at sites that are freely accessible and at places that you're not going to potentially disrupt landowners and such. And so the methods, yet again, we were still using the North American bat grid that we had already implemented. We had the information from the Texas Department of Transportation, as well as known information of historic tricolored bat colonies based on previous survey efforts. And from all of that, we had surveyed 208 culverts in Texas. And when we came to a site, we always first assessed whether or not we really wanted to go into it. These sites tended to be fairly short, about 1.3 to 1.5 meters on average. And when we got up the courage to crawl through these, we recorded the length of the culvert, the height of the culvert, the width of the culvert. We also recorded some culverts that had multiple sections or just multiple culverts stacked next to each other. So this image is showing one that had 5 different sections. We also recorded aspect, so the directionality of the entrances, whether or not it was more northern eastern or northern southern and eastern western facing entrances. And we also documented the presence and distance to water, as well as the percent portal obstruction and the elevation of the site. And we pulled in some information such as external temperature from the Prism Climate Group, vegetation index, so just how green was it at these sites, and it's a percentage, and also the vapor pressure deficit externally. Once again, just another way to kind of get an idea of the humidity. And so of the 209 culverts that we surveyed across Texas, we identified 49 culverts occupied by tricolored bats, and we counted 3,375 tricolored bats. And in addition to tricolored bats, we also documented a couple cases of the big brown bat as well as the southeastern myotis. And I won't get into the statistics of it all, but using all of those variables, we found that predicting presence of tricolored bats was impacted by the greater length of a culvert, the size of the portal opening, lower vapor pressure deficit, meaning more humid, and higher temperatures. And for abundance, the one factor that seemed to be driving predicting increased abundance of tricolored bats was the number of sections. So with more area, bats might be occupying more space. So to discuss this, we found that culverts in areas devoid of caves can be really important sites to consider as potential habitat for bats such as the tricolored bat. We found that predictors of presence and abundance of tricolored bats did differ. We can use some of this information to identify other potential culvert roosts within the known range of tricolored bats. So tricolored bat range in Texas is nearly statewide, but not quite. And from previous notation, the Texas Department of Transportation had more than 60,000 different features. And so using these variables, we can then constrain and sort for certain ones that have a higher probability of having this species present. And of course, this was just important because we started having this threat of white nose developing in the state, and we knew that the tricolored bat was most likely going to be impacted. And in addition to that, it had been petitioned to be listed as a threatened or endangered species. And so having all those tricolored bats occupying culverts kind of became the focal point. And from this study, There are currently ongoing studies in collaborative efforts with the Natural Resources Institute, TPWD, as well as Kennesaw State, looking at different ways of managing those tricolored bat populations in those culverts in Texas. And finally, we come to kind of one of the last projects, the big project that kind of sums up the whole study in a way, and that is to model the predicted spread of white nose in Texas. So unfortunately, in spring of 2020, we had documented, Texas Parks and Wildlife Department documented the disease in Central Texas. So with this knowledge and with the knowledge of the distribution of different caves in Texas and all of the different bat characteristics that we had collected and the environmental characteristics, so using those data loggers we had deployed in caves as well as external temperature, we are now working on trying to develop a potential spread of the disease just for cave sites. And we're focusing just on caves to keep the overall model as simple as possible. And in addition to that, we're trying to keep it general to just bats in general, rather than looking at different species, because there are so many different species, and that can make a model more difficult to interpret. So at this point in time, we are currently working on writing up the results of the model and trying to get some predicted outcomes so that these different figures can then help, in addition to all the other information gathered, try to hone in on different regions in Texas that should be more focused on for obtaining access to sites for management of the disease, and to try to get an idea of what might happen 5 to 10 years out should this disease continue to develop and spread in the state of Texas. And so overall, the benefits of this entire study, not just the model, but all the previous studies and all of the information that was gathered, this all can benefit future projects. So as I'd said, we had a lot of information that had been collected over the years by cavers with their known knowledge of a lot of caves and bats occupying those caves, other researchers in different regions having different information. And so this study kind of combined all of that knowledge into one updated database. And in addition to that, with all of these survey efforts, we were able to track the spread of the fungus through the state of Texas. And also then with that information, the identification of white nose syndrome, which happened in spring 2020. And this can all lead to just general management and conservation efforts for different species of bats in the state of Texas. And so this study was entirely very important for just understanding everything that was currently happening. And in addition to those other studies, we had several others that were published off of this information, and we're still working on other publications. And with that, I'd like to acknowledge everybody in the state of Texas for their assistance with this project. We had so many different collaborators and so many different funders, and the entirety of this project would not have been possible to the extent that it was with all of this different assistance. And also, of course, you have to have great field teams that want to crawl through bat guano under roadways to collect all of this data across the state. So with that, I'd like to say thank you and kiitos from Finland, and I will take any questions.
Thank you, Missy, so much for that that presentation. Looks like we just had one question come in. It says the instances of PD, that's the fungus that causes white nose syndrome, seem to be concentrated in the eastern panhandle and central Texas, although many bat roosts were also identified in East Texas. Do you have any ideas why?
That's an interesting question. I've got to say that it's really difficult because the fungus can spread bat to bat. And so we definitely were not biasing our sampling efforts in caves versus culverts, but obviously our sample efforts were 25 bats per site, and we were finding this fungus at such low quantities. In addition to that, we had different kits, and so the methods may have provided different results. But in general, the fungus is at such low quantities, the DNA, we might have just missed it in those eastern sites, especially in those culverts. But in general, I can't say whether or not any of the sites were different enough that we wouldn't have picked it up eventually in those sites.
And I want to piggyback on that one. We have several other questions that just came in. The only other thing I would say to that is, I think the Eastern Panhandle ones made sense because initially they were the closest sites to where PD was before, and so that one was obvious. The Central Texas one is a little bit trickier to explain. Clearly, there's a bunch of caves there, and so that might be what's going on. So then the next question is, it says, hi, I am an undergrad pursuing civil engineering. I'm interested in learning more of the bats here in Texas, but just wondering if the bridge designers don't mind having bats make habit or I guess probably start making use of the bridges, or is it something they look to purposely design in a bridge? I guess it says in a WA. I'm not sure what that means, but I think they're just wondering about, do the bridge designers mind having bats or yeah.
Yeah, so when I was working with the Texas Department of Transportation, I even came across bridges where they put up bat boxes. So it kind of just depends on, what's happening. Obviously, as a engineer working with bridges, if you have to modify or do any construction, it's difficult when you have a species of bat or other species of wildlife occupying the site. But I think in general, you're not working on those sites, they're good to have. I never came across any transportation folks that did not have issues with, there were no issues with the bats. And they actually, worked really well with the biologists. They were really interested in the bats. And I know that there was a study by Keeley and Tuttle some time ago that were identifying these different characteristics, and that some bridges were purposefully designed to attract bats to them.
There was a question about what precautions we took to disinfect our team to prevent the spread of white nose syndrome, and I think that's a pretty quick answer, probably. But yeah.
Yeah, so thankfully I didn't track myself spreading the fungus. Between sites, we wore these coveralls and we had our helmets and our everything on. We would bleach everything between sites and wipe down everything with Lysol wipes. So we were following the white nose decon protocol at every site.
And then we had a question, has it been confirmed that Tadarida, or the Mexican free-tailed bat, can't harbor the fungus at all, or can they become a spore carrier to other species that may be sharing a roost with them?
Yeah, so we did document that the Mexican free-tailed bat did have presence of the ticks on it, no signs of white nose. It is thought that this species might not be affected by the disease because they're not known to hibernate or go into that torpor period for long periods, which is kind of necessary as the bat's immune system is downregulated, so they can't fight off the disease. And so at this point, this bat is in fact potentially a carrier, and I did document this species occupying different caves where other species were present, and so it can spread it obviously down into Mexico, other sites in both northern, eastern, and western regions of Texas, as well as outside the state of Texas.
Then there was a question, is the evening bat being looked at for vulnerability? This is a very common species in north central, northeast Texas.
Our team never documented the evening bat. I am not sure at this point what studies are going on. I know that Texas Parks and Wildlife is funding a project that is looking statewide at bats and the impact of white nose. However, I do not know if there's anybody specifically trapping this bat and swabbing it. So if we were to get any samples for my project, it would have been from wildlife rehabbers.
Yeah, I mean, yeah. I think our hope is that species is less likely to be susceptible because it's not a big winter hibernating bat in big roosts like some of these other species. Okay, lower temperatures are required for white nose syndrome to survive. What about transmission? Does the temperature also have to be low? I'm thinking about Mexican freetail in Mexico. Like, if they had the fungus in their body, could they transmit it in a warm environment, I guess?
I mean, I guess it depends on what temperatures you're looking at. If they get too warm, obviously you're probably not going to see any fungal growth, so you're not going to get sporulation, less likelihood of spreading at a certain temperature. I'm sure the fungus just does not sporulate, and then you don't really get the spread of the fungus, and you might not even, at that point, you're not going to get the development of the disease.
And Mike Yanish asked if all the sites were surveyed every year.
No, so we did our best to try to resurvey we actually did pretty well resurveying sites that we knew had bats, because those were obviously highest priority to resurvey those. We had a lot of sites that didn't have presence of bats, so we just surveyed them once and, went to the next site. And that was just to try to get more of a statewide idea of what was happening. So we had to kind of cut our losses on repetitive survey efforts at certain sites and just focus on the ones in that had bats yearly.
And then there's a question about, could a projected spread of PD influence a listing decision for a bat species?
I would hope not, because the projected spread is obviously just a model. Models don't tend to be very accurate, but they can point out different parameters or features, such as hibernation period or species, that could influence the rate of spread of the fungus in the state. And so the idea of the model is more for just identifying those features and kind of possibly looking at different regions. But beyond that, we're taking a very general approach and not looking at any specific species. So in the case of the model we're working on, there would be no way to use it to list any specific species.
And then what advice, lessons learned, or improvements would you recommend to someone wanting to do a white nose syndrome assessment in their state?
Ooh, it depends on the state. So I found out real quick that getting access was the most difficult part. And so throwing yourself into any possible avenue of gaining access. So, if you need access to caves, cavers are really like saving grace. They tend to have the access. They've got great landowner relationships for the caving environments. Talking to other biologists throughout the state. I mean, it's really about getting your information out there. In Texas, we also sent out holiday cards to keep people apprised that we would continuously be coming back to their property to survey. And so, I mean, I think the biggest way of doing this is to figure out, all the potential hibernacula sites, get all that information, and then just trying to get that access is key to starting a study like this. And then it really just also helped that we had so many different groups, such as Dr. Winifred Frick and National Wildlife Health Center, that were also interested in looking at multiple different states for swabbing for this fungus. And so that kind of reduced some of the costs on swabbing kits and effort.
So the next question I'll go ahead and take, it was, did PD spread to Texas bat populations by contact from other bats, or did it spread by humans? And what we saw was a very gradual, predictable progression and sort of geographic uh, spread of the fungus that indicates that it was spread by bats. We didn't see any big, giant geographic jumps, which would be a signal of the potential for a human spread. There's no way to know 100%, but it really did seem like it was a very gradual progression just along the disease front, right where we'd expect it to spread. The next question was, what type of PCR was run on the extracted DNA? Do you know if anyone is using droplet digital PCR since it tends to be more sensitive?
Oh, well, I can't recall off the top of my head. So I don't know anything about the droplet PCR in particular. And I knew that the labs had a specified PCR methodology that they were all utilizing. Unfortunately, I can't really give detail about it myself.
And we have, let me see, just like a couple more questions here. Let's see. It says, thanks for a great talk. Could you elaborate a bit on the predictors being considered in the infection model? Also, are there predictors that you thought would have been included that are actually not as informative as you would have expected?
That's a good question. So we're still making modifications to the model itself. Some of them, one in particular that I can recall off the top of my head was just the hibernation temperature. So a lot of the predictors we're seeing were fairly, I don't know, they made sense for what is driving the development of the disease. And because we had this knowledge of where the disease has already been shown, we could kind of tailor our model to kind of point that out. And so the environmental temperature, hibernation, duration are kind of some of the key parameters that we're seeing thus far as drivers for the model.
And then we'll just take one last question from Nick Fuller. He says, can you describe the spatial distribution of bat roosts in highway culverts?
Spatial distribution of bat roosts and highway culverts. So that's kind of easy for the culverts that I surveyed. The majority of the bats, which tended to be the one species, the tricolor bat, they were occupying culverts along the I-45. So for any of you in Texas that are commuting between Houston and Dallas, you are driving over different colonies of tricolor bats. We also found a couple colonies more farther out east, but predominantly in that eastern region. And we found a few others kind of scattered in central Texas as well.